Yes.
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 Yeah.
Okay, so what I thought I would do today is give you a little bit about the Pfizer and Moderna mRNA vaccines, which I actually started preparing this before the Omicron arrived, but it's probably even more relevant now. So first of all, what are these vaccines? And then I will do a comparison. So each of these mRNA vaccines didn't sort of come out of the blue. They reflect decades of research on the nature of RNAs. So here's the basic idea. Let's see, can you see my pointer?
Yeah.
Yes.
Okay. Here's the basic idea. And there is a base sequence, which is simply a set of nucleotide bases in sequence. And in combination, they encode a messenger RNA. The messenger RNA is enveloped in a lipid coating, because that is needed to get the messenger RNA into the cells, and that is what is injected into the body. So here's your messenger RNA. The lipid enables it to get through the surface of the cell membrane. The mRNA is then exposed and goes to a little organelle called the ribosome. The ribosome translates this messenger RNA into proteins, and it encodes for the spike protein of the virus. And this then goes in two directions. The spike protein is displayed on the surface of the cell, and that is detected by one of the cells of the immune system that then reacts to it. And another one, the spike protein is broken up into little parts, and little parts get given to another cell of the immune system. And here's what the virus actually has inside it. This is the basic idea of the messenger RNA. It's in this capsule. I got to move my own. But it's in this capsule, and these little squiggly things are fatty acids. One end, the little ball end, is hydrophilic or water-loving, because mRNA is soluble in water but not in lipid. And these squiggly tails represent the hydrophobic or more fatty part of the fatty acid. And this circle here shows you that this hydrophilic part on the outside makes it the whole vaccine water soluble, but it enables it to get through the cell membrane. And the basic part here's the vaccine. It consists of parts at the beginning and end that improve its effectiveness. And then in the middle, it has the gene for the spike protein. On the end, they also stuck on polyethylene glycol, which is the same thing as you have in your antifreeze in a car. But that also assists the solubility of the whole, what is called a lipid nanoparticle. Lipid means fatty, nano, very small particle. Okay. I gotta move this so I can. I don't know what you see, but I part of the Zoom overlaps it in mine, but I hope you can see it. Okay, here's the parts of the actual vaccine. So it's a fairly complicated structure. I got to just turn off a junk call. First of all, there's a small cap unit which protects the RNA from degradation. There's an untranslated region, UTRs means untranslated region, that causes high levels of translation from the correct starting point. This enabled SIG is a signal peptide. A peptide is a chain of amino acids, and that guides the actual mRNA into a membrane system within the cell where it can produce the correct spike protein. The middle part is actually the instructions to make the spike protein. And it's not just the mRNA reflecting the spike protein. It also contains a couple of purposeful mutations in it, which ensure the correct three-dimensional structure of the spike protein to fuse with the cell membrane. There then follows another untranslated region, which enables high level of expression and stabilizes the messenger RNA. And finally, at the end, there's a polyadenosine tail. All messenger RNAs have a polyadenosine tail, but this is a rather unusual one with extra, it's rather large and has extra stretches that they've put in it, which increase the stability of messenger RNA and also preserve its correct structure during the production. The production is an enzymatic production because the whole thing is too big to do by standard chemical reactions. Here's the sequence of all the bases in it, and it is over 4,000 bases long. Here you can see, for example, the poly-A tail. Now, there's one other interesting thing about it. Normal messenger RNAs contain 4 bases: adenine, guanine, cytosine, and uridine. But the messenger they created does not contain uridine. It contains a different base called methyl pseudouridine. And the reason for this, it turns out that when they were working with trying to get these expressed in cells, they found that uridine increase or produces an immune reaction in the cells. But if they use this derivative of uridine, which works exactly as equally well in producing the required protein, it reduces the immune reaction of the cells. So that's the entire set of instructions. That is actually the Pfizer vaccine. And I will show you a comparison of those two sequences in a minute if I can get it up. But in addition to the messenger RNA, there are these lipid nanoparticles I spoke about, and they probably determine where the mRNA ends up. And they are not quite the same, they are similar. And the top, this is a chemical formula, which I won't bother to explain at the moment because we'd have to go into a little bit of organic chemistry. But let's say this is a diagram of the lipid that's in the Pfizer vaccine. And this is a diagram of the lipid that's a lipid nanoparticle in the Moderna vaccine. So not only are their sequences slightly different, they are in slightly different nanoparticles. And the Moderna vaccine has a higher level of mRNA in it than does the Pfizer vaccine. So that's that's where I stop sharing that, and I'm gonna try and do one more thing for you, hopefully. Now, no, I hit share, but that's sharing the wrong thing. Now I'm gonna try. I'm going to try again, share screen. And if here's the screen I want to share. Okay, can you see that series of letters on the screen now?
Yes.
Yes.
Okay, on the top, these are the series of letters this is actually the DNA template on which the messenger RNA is made. The top.
A quick interruption, Peter. I see that there's timing now, and there wasn't before. In your earlier slides, you had something, it was uracil.
Oh, it was uridine. This,
Uridine.
yes.
This is DNA sequence.
This is the DNA sequence that is used as a template during the manufacture of the messenger RNA. And this is what I copied from another paper. And then what I did is I copied these sequences and fed them into Word and asked Word to give me the comparison. And so this is what Word produced for me. And as you will see, the Pfizer is on the top, the Moderna is on the bottom. And in general, if you go along and compare these, they are pretty much the same, except sometimes they are not. Now, this, without dividing it all up, the three of these bases encode each amino acid, and sometimes a group of three may have some redundancy. So I cannot tell you without spending hours on this, whether this makes any difference to the protein. They may be just different, what they call groups of three or codons that produce the same protein, or they may be slight differences in the protein. But here we go all the way along. I've highlighted some in yellow that I found where they're different. As you'll see, they're mainly the same. I didn't go all the way through. But here's the entire sequence. Whoopee, whoopee. I think we've done enough for you to see that they are basically identical, but with small differences. So these small differences and the different lipid nanoparticles probably account for the differences in the slight differences in the effectiveness of these two viruses. Now, once they, as they already have the sequence for the Omicron variant, I think these pharmaceutical companies can very rapidly stick that variant in the same lipid nanoparticles and produce the vaccines, I would say, in very short order. They don't even have to go through the same level of trials because the immunogenicity or the reactions of the body would be very similar. If you change a few letters here, it's not, in my opinion, going to make much difference. But
Peter,
a slightly different spike protein. Yes, Kirk?
One question. So this is the E. coli DNA sequence that codes for the mRNA?
They did it in plasmids. I guess they would have done it in E. coli. I
Yeah, I
didn't.
think they grew it in E. coli.
Yeah, okay. Well, this would be the sequences in the plasmids in the
Right.
E. coli. Yeah.
Okay.
And they were produced as a vast chart in a PDF, and I had to copy it and put it in Word, and then I asked Word to do a comparison, which actually worked pretty well. We got all 4,000, and we can even go right up to the far end, I think. And you can see the poly A tail. Oh, maybe you can't. I thought it would go to the end. Anyhow, I think you've had enough of that. Anybody got any I'll stop sharing. Anybody got any questions on that? Yes, Ed. Ed, you're muted.
You didn't mention antibodies. What are antibodies?
Oh, okay. When I'm not an immunologist, and maybe Kirk can even tell me more about this, but when the cells of the immune system see these spike proteins, they recognize them as foreign and produce other proteins that sort of glob onto them and prevent them from, when the virus comes, they glob onto the virus, the antibodies and other proteins, and prevent it from getting into the cells. And if you do that, you are immune. Kirk,
Okay.
The antibodies are kind of Y-shaped molecules that recognize a specific protein or other chemical moiety and attach to it. And then when there's an antibody antigen complex, that stimulates all kinds of both raw chemical reactions, various cytokines, and it also stimulates a variety of macrophages, cells that recognize and then digest those. So the antibody that is produced when you flood the bloodstream with the spike proteins coming out of the cells, the immune system cells see that as a foreign protein and generate the antibodies, the antibody response to that. And so if there's a change in the spike protein, enough of a mutation to that spike protein, then the antibodies won't recognize it as foreign and won't attach to it. And so that's when you might need an alteration in the vaccine, an alteration in the mRNA to produce a spike protein that looks more like the new variant.
Okay, good, thank you.
Okay.
That sound good, Bill?
Yeah, I gathered from what you presented, Peter, that the actual protein is expressed on the surface of the cell that the vaccine has penetrated, and that spike protein on the surface of the cell is what the B cell is recognizing and then generating antibodies. Is that correct?
Well, that's what I understood from that diagram. And also a part of it is also presented to the T cell, but I'm an immunologist, and I got the impression it was the two together, but these are presented on the surface of the cell for the cells of the immune system to recognize. And the immune system cells are circulating in the blood. Is that correct, Kirk?
Yes. The thing I don't know is the mRNA nanoparticles, is that taken up in the local muscle tissue, or does that enter into the bloodstream and then taken up by other cells throughout the body? And I don't know the answer to that
No,
question.
I don't know the answer to that either.
No, I wouldn't be able to say specifically, but I would wager, I would put money on the fact that it's actually circulated and then reacted to.
Yeah, that's what I've assumed, but
Yeah,
I had
I
that
would
thought.
think it's more likely than the muscle system
Yeah,
cells.
The importance of the T cells, which is a separate system of immunity, is that when they are looking at this Omicron virus and they're using the antibodies that they have from vaccinated individuals to test against the virus, all they're looking at is the B cell antibody immune response. And that's not the whole story. So let's say there are, I guess, a couple of different outcomes we could anticipate. One would be, yes, the antibodies tend to be effective against Omicron. And so we can say the vaccines that exist are giving good antibody protection. Or the outcome might be not so good. A third outcome would be, doesn't look like it's effective at all. But that doesn't take into account the T cell system and its ability to react to the Omicron virus. And so the story may not be as dismal as it could be if we find that the antibodies of vaccinated individuals don't neutralize the virus as effectively as we desire, because we still have this backup T cell thing going for us. I can't really tell you exactly how effective that is compared to the B cell antibody response and what that means about severity. So that's beyond me, but that's part of the important dynamic that's going on here.
So Mark, as you might be in charge of speakers next year,
Yes,
interesting to see if we can find an immunologist to give us immunology 101 in 30 minutes.
good. I will put that on my list of possibilities here. Thank you for the suggestion.
There must be 2 or 3 immunologists at Cornell
Right.
could give us a basic explanation.
The other thing about the development of a vaccine, let's say that it does become necessary to have a vaccine targeted to Omicron. The latest I heard was that probably a couple of months would be required to be able to really develop it and then ramp it up to being a meaningful production for use. So it wouldn't be like we can turn on a dime. It still takes a painfully long time for it to be developed and then distributed.
yeah,
Yeah,
you'd have to figure out the amino acid sequence of the spike protein, and then the differences, and then reverse engineer a new messenger RNA sequence to code for that. And then you've got to make the E. coli plasmid DNA and get a bunch of E. coli, get that into an E. coli cell and have it multiply enough to generate the messenger RNA. And then I guess they cook it in vats and just have all these little bacteria churning out messenger RNA molecules.
but the fact that they've already detected all the mutations, they probably know the complete DNA sequence
Right.
of the virus. I don't know, it might be an RNA virus. I'm not quite sure, but
It is.
they've probably got that already. So once you have that sequence and what they know already, they know which part of the sequence encodes the spike proteins, and they just reproduce that.
Okay.
There is also the antibody treatments that they're using. And I assume they're looking at that to see if those antibody treatments are effective against the new variant.
Good point.
Well, Kirk, I did have a whole lot more, but it is 2:00. So
Okay.
How
I
about
will-
do,
I
how
will
about continue next week?
I
And
will
continue-
put them for a future time.
Right. Well, yes, the antiquity presentation should be of interest at some point in the future. We're looking forward to that.